folks, and welcome to Anatomy and Pathology on Demand, where we are finishing week 6. This is the fourth and final lecture on tissues. So let me go ahead and get the slide up, and then we can get started. So far we've talked about tissues in general. I mentioned that there were 4 types. We talked about connective tissue, then we talked about muscle tissue, we talked about nervous tissue, and now we are going to talk about the 4th type of tissue, which is epithelium. So here's the thing about epithelium. It's cell, 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 followed by cell, 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 cell. In other words, no extracellular matrix. It's just rows, sheets of cells. And these sheets of cells cover every single surface in your body. And by every surface, I mean everything on the outside, every surface on the inside. So inside your mouth, on the inside of your eyelids, inside your nose. It lines the inside of all your blood vessels. It lines the inside of your stomach. There's a lining of epithelium on the outside of your stomach. It also forms all your glands, because if you think about it, glands are like little tubular structures for the ducts, and then they open up into a bigger cavity, meaning a space. If it's a space, it means it has a surface. So if it has a surface, it has epithelium on it. Okay. The unique thing about epithelium is it's just rows of cells. So there are no blood vessels in between any of these cells. So it is what we call avascular, meaning there's no blood supply to it. However, there is a nervous supply. So there are little nerve endings that come up to it, and there's little sensory receptors from it. And we'll learn more about those, for instance, when we learn about the skin, which is the covering, which is going to have an epithelium in it. And as opposed to a sarcoma, which was a malignancy of all connective tissue, a carcinoma, that word carcinoma, if you've ever heard basal cell carcinoma or squamous cell carcinoma or adenocarcinoma, that word carcinoma is telling you it's a malignancy of the epithelium. And the unique thing about epithelial cells is they turn over very quickly. So the cell cycle is shorter compared to the cell cycle of some other cells. And as you learn about various organ systems in your body, you will talk about how quickly the epithelium can turn over in those places. So because the epithelium lines such a diverse areas of your body, there are multiple functions to the epithelium. Protection is the main one. Your skin keeps stuff from entering your body. You have to have a break in your skin for something to actually enter your body. Remember, there's no blood vessels in the epithelium, so that little microbe cannot get inside your body until it has gone through the epithelium and gone deeper. So it's forming this barrier to keep stuff outside, to keep you healthy. And yet, epithelium has some characteristics which will allow some things to enter the actual cell, so some stuff can pass through it. And as we all know, some epithelium has features which allow it to secrete. That's what your runny nose is. The epithelium inside your nasal passages have certain cells that have secretions. And it also can absorb substances. And I don't really mean like Easter egg dye, but like, for instance, if you've ever heard of people taking the nicotine patch or a birth control patch, is because they can absorb the medication through their skin. So when we look at epithelium, there's a lot of special characteristics. So let's first look at the cartoon drawing on the left, where you have 3 yellow cells all lined up next to each other, and we have intercellular junctions, which we talked about previously. If you notice, the cells are sitting on the underlying tissue. So that deep area is called the basal part, whereas the part on the surface, since epithelium lines the surface, there always has to be a surface part, and that area is going to be called the apical region of that. And so the fact that it has an apical side and a basal side, that is what we call polarity. I already talked about how it's avascular, but has nerve supply, or it's innervated, and how quickly it turns over. There is a structure underneath these cells of the epithelium called a basement membrane, which I will talk about in a moment on the next slide because I have more details. And this stains distinctly. So the way you can tell you're looking at epithelium in every single slide is you're going to have a white area. You're going to have an area without side, anything in there. That's showing you have a surface. So if you have a surface, you have to have an epithelium. And then in this one, you see this dark pink line here where the arrow is? That is showing the basal aspect of that epithelial cell where we have that basement membrane going on. And so the cells are going to be attached to each other on the sides, and they're also going to have attachments down there at their basal regions to the basement membrane. So let's look at that basement membrane a little bit closer in this drawing. So here we have 3 tall, skinny epithelial cells, and then the blue part underneath it, that is meant to represent an areolar connective tissue. And between the two, we have these brownish areas. Now, technically, there are 2 parts to this basement membrane, something called the basal lamina, something called the reticular lamina. But for this course, we're not going to use those 2 distinctions, and we're just going to go with the word for the entire structure, which is basement membrane. So if you can remember that a basement is deep to a house, then the basement membrane is deep to the epithelial cells. Okay? And so these cells of the epithelium are anchored to this basement membrane. And so it's kind of controlling what's going through the cells, has to go through the basement membrane in order to find the nearest blood vessel in order to get into the body. When we look at this basement membrane, part of it is secreted from the cells above, part of it is secreted from the connective tissue below it. And there's a lot of protein fibers in there. And so one structure that attaches the actual cells of the epithelium to the underlying basement membrane is something called a hemidesmosome. So if you go back to when we talked about the intercellular connections, where we talked about the tight junctions near the surface, and then we talked about the desmosomes or the anchoring junctions here, which were holding the 2 cells together so they couldn't be ripped apart. A hemidesmosome is like half of an anchoring junction, and the other name being for an anchoring junction being a desmosome. Hemidesmosome literally means half of a desmosome or half of anchoring junction. So it makes sense. If you don't remember what anchoring junctions or desmosomes and gap junctions and tight junctions look like, make sure you take a moment and review it in your textbook. McKinley has a good description of them. So here is what the hemidesmosome at the bottom looks like. We have this piece of protein within the cell, and then you have protein elements like half of a zipper, which is attaching it to the underlying basement membrane. That's what these little lines are. And then coming out of this protein plaque, these red fibers, these are big pieces of keratin, which is one of the types of protein fibers that go into the cell, which basically, if the cell is trying to pull off the basement membrane, the tension is going to be relayed to the upper areas of the cell. So it's kind of like if you think of a telephone pole with the guy wires, attaching it to the ground further out. When the wind blows and it wants to pull, it's going to have to pull that guy wire as well before the telephone pole can fall over. So that's what those red keratin filaments are trying to show you. So it's attaching the basal aspect of epithelial cells, the ones sitting right on the basement membrane, is attaching them to the basement membrane to resist any kind of shear force, like friction when you're rubbing things together. All right. When we look at epithelium, we classify it, and we classify it by looking at 2 different things. The first thing we're going to look up is what is the shape of the cell? And here we have 3 choices. Is the cell mostly flat? Okay. super skinny. Maybe the nucleus sticks up just like the yolk on a fried egg sticks up. The nucleus is sitting in the middle. It's usually round. This is going to be our thinnest cell, which means substances can diffuse through it very quickly. And that type of a cell is called a squamous cell. The next cell shape is we're going to make the cell a little bit thicker, and we're going to make it boxy. So it's going to be like a square in 3D, which is a cube, and this type of cell is called cuboidal. And in the middle of the cuboidal cell, you're going to have a round nucleus sitting right smack dab in the middle. Now, on cuboidal cells, you could have microvilli to increase the surface area. And if you remember what we learned about microvilli, we would have them if that cell is going to be secreting or if that cell is going to be absorbing, because the whole point of the microvilli was to increase the surface area. And the third cell type is we're now going to make the cell even taller. So it's going to be long and skinny, kind of like a column. So we call it a columnar cell. It's much taller than it is wide. The nucleus can either be round or it can be oval. It can be sitting near the basal aspect of the cell, or it can be sitting near the middle, but it's going to be in the bottom half of the cell. These cells can also have the microvilli to increase the surface area, in which case they're either going to be absorbing or secreting. And in addition, these cells can also have our friends, the cilia, to create external currents to move things along them. And in fact, they can have both microvilli and cilia. So cell shape, squamous, cuboidal, columnar. That's the first way we classify it. The second way we classify as epithelium is we look at how many layers there are. We find the basement membrane, or we count how many layers up to the surface, or you find the surface and you count how many layers until you hit the basement membrane. If there's only one cell layer, we call it simple. So if it's one cell layer of a squamous cell, it'd be called simple squamous. One cell layer cuboidal would be called simple cuboidal. One cell layer columnar would be called simple columnar. So you always want to give multiple words when you're naming the type of epithelium. And so simple squamous would be the thinnest of all the types of epithelium. So this is the one where we want stuff to diffuse really quick when we're osmosis for water movement, or secreting and absorbing like oxygen, carbon dioxide. We're going to have a simple squamous epithelium. All right, if we're done counting and we can continue on, whether we can count to 2 or 20 or 200, we're going to use the same word, and that word is stratified. In other words, more than one cell layer. Okay. And what happens is the cells are first made down by the basement membrane, and then they're going to move their way towards the apical surface. So the cells by the basement membrane are the more immature cells. We are not going to use their cell shape to classify the epithelium. Instead, we look, what's the shape of the cells near the apical surface? Are they flat? Are they square? Are they tall and skinny? And so then we would call them stratified squamous, stratified cuboidal, or stratified columnar, respectively. So the reason why we have a stratified epithelium is because we know there's going to be some type of trauma to it. With a simple epithelium, if you rub against it and that one cell's gone, well, then the epithelial surface is gone. And there's no more protection going on. So in high abrasion areas, we're going to have stratification to protect the underlying tissues. Now, there is something that I like to call the great anatomy student fake out. And that is something at first glance looks stratified, but if you look at it closely, you'll realize it's not really stratified. So it's a fake stratification. It's what we call pseudostratified. And so for instance, if you look at this picture, what you will notice, like at first glance, it looks like we have nuclei all over the place. But what you would notice looking at this is we don't have a row of cells with nuclei at the bottom, and then another row of cells with nuclei higher up, and another row of cells with nuclei higher up. In fact, what we have are cells that are going all the way from the basement membrane all the way to the surface, and it's just the nuclei are all over the place, okay? And remember, in columnar cells, the nuclei are supposed to be in the bottom half. So if you see nuclei in the top half of a cell of a columnar cell, it's going to be this fake out, okay? Because pseudostratified is always columnar. You don't have pseudostratified, squamous, or cuboidal. So you wouldn't say simple cuboidal, you would say pseudostratified cuboidal. And here's another clue. Pseudostratified cuboidal epithelium 100% of the time is going to have cilia. So if there's no cilia, it's not pseudostratified columnar. And the other thing is almost always pseudostratified cuboidal epithelium are going to have what we call goblet cells, which look like these little champagne goblets, which is this green cell over here on the left side that I'm pointing to. And what these are is single cells that secrete mucus. I'll be talking more about those in the lecture for Monday of week 7. So come back and pay attention to that again on Monday. So let's put these things together and look at various epithelium and talk about where we might find them in our body. Oh, but before we get to that, let's talk about what happens with the tubular structure when you cut it, how it changes, how it looks. So if we have a long cylinder and we're cutting it on the transverse plane, we call that a transverse section. And so you're not going to get the idea that it's long and skinny. You're just going to see something that looks kind of like a lifesaver, where it's got a hole in the middle and then we have something on the outside. Now, if we take that same tubular structure and we cut it in a sagittal plane or in a coronal plane or a frontal plane, what we're going to see is the lumen in the middle between the two, and then we could see epithelium on both sides facing where the lumen is. And lastly, if we cut things at an angle or an oblique section, it's not a perfect cross section, so it looks kind of oval. So it's slightly distorted. It's kind of between a cross section and a longitudinal section. And so this explains why when I'm showing pictures of histology, they're not all perfect little longitudinal sections or cross sections, because we just cannot line up our slice with the anatomy. Okay. So simple squamous epithelium, the thinnest of all the epithelium. The top picture shows what it would look like from the side. The dark purple is the basement membrane, the yellow is the cell, and you can see the little squished nucleus. If you were to look at it from above, the picture below shows you what it would look like from above. It's like this whole pan of fried eggs sitting next to each other. So here's simple squamous epithelium in 2 places in your body. The one on your left, you see how thin it is and see how the bulge is actually the nucleus sticking out? This is what it looks like in normal airways. This is your lung, not airway, sorry, and the parts of your lung where we're doing gas exchange, a structure called the alveolus that you don't know about yet. And so it's super thin, so the gases can go into and out of it very quickly. On the right, conversely, this is from your kidney, and the part highlighted in green is showing the simple squamous epithelium. So you can also see how flat it is. And remember, in the kidney, this is where we need to have fluids and ions and things get through very quickly. So simple squamous epithelium, we're going to find when we need to filter or have things diffuse very quickly. So it's also found on the inside of blood vessels. And even though it's an epithelium, because it's on the inside of blood vessels, we're going to use the word endothelium for it there. But you won't be dealing with blood vessels pretty much until the 2nd semester. Moving on to stratified squamous epithelium, where it's labeled basal lamina, you can just scratch it out and write basement membrane. So that greenish part is the basement membrane. And as you can see, we have multiple layers of cell going above it. Now that deepest layer here definitely looks cuboidal, but remember we name it from the apical or the surface layer, and the surface cells are flat, and that's why this is stratified squamous epithelium. Realize in a stratified epithelium, only these deepest layers are where we're having the cell cycle mitosis, and then they are moving upward and they are dying as they move closer to the surface. Now if you notice, these two pictures don't look quite like each other because the one on the right has all this yellowish stuff on the top. And this is the difference between mucosa versus skin. So the one on the right is the one on the right over here. And this is what skin looks like. Okay, so the outside of your cheek. Now, if we take that same epithelium and we put it on the inside, it's not going to have this waterproofing outer layer to it. And so it doesn't have, if I go back to the previous picture, it doesn't have this stuff on the outside, which is called keratin. It's naked for the extra layers of keratin. And so the inside of your cheek is the exact same stratified squamous epithelium without that extra keratin. And that is the picture in pink on the left. And that is called a mucosa. So epithelium, like on the inside of your digestive, respiratory, reproductive tract, urinary tract, that's going to be called a mucosa. So skin is what we call keratinized because we have these layers basically of completely dead cells, and just the keratin that they produce are on the top, and it's very protective to us. Whereas mucosa is not keratinized. So if you have decided a certain tissue is epithelium, and then you have decided it's stratified squamous, the last thing you have to decide is it keratinized or is it not keratinized, stratified squamous epithelium. And mucosa, esophagus, inside of your mouth, your vagina. And here is an easy way to tell, like if you're worrying, is this keratinized or not? If you've decided to stratified squamous, look to see if you can see nuclei going all the way to the surface or almost to the surface. That makes it mucosa. When you look at the one on the right, you can see that the nuclei kind of die out in this area. You can tell the cells have flattened. So it's going to be squamous, but you don't see any nuclei up here, just like you don't see any nuclei up here, because that's all dead. It's just bags of keratin up there. All right, done with squamous, let's move on to cuboidal. So in cuboidal, the drawing on the left is showing a flat surface, a simple cuboidal, whereas the one on the right is showing a tubular structure like in a duct or a gland. This is the cross section, just like this bottom picture is the cross section, where this darker gray line, which I'm going around right now, this is where the basement membrane would be. And then here's an adjacent duct and it would have a basement membrane. So right here, this white space in the middle, that's the lumen. So you can see that there's only one cell thickness between the lumen, the apical surface, and the basement membrane. Looking at the purple picture on the right, these are longitudinal sections going through the duct. So at first glance, you might think that this is two layers of cells, but it's actually not. Here's the basement membrane for this duct, and this is the basement membrane for that duct. So it's actually two ducts right next to each other, which have been cut like this. So both of these are simple cuboidal epithelium. You can see a nice large round nucleus in the center of each of these cuboidal cells. So as I went along, I said what type of sections these are, but looking at all these pictures today, go back to where I talked about the three different types of sections and decide what type of sections are being shown. So the simple cuboidal epithelium is seen in a lot of our glands, as I alluded to, as well as tubules that we're going to find in our kidney in the second semester. Stratified cuboidal epithelium also occurs, and at the top is the flat surface drawing showing a cuboidal followed by cuboidal on top, but realistically it doesn't usually look like that. If we look at the cross section down below, you can clearly see that it's two cell layers. This whitish area, that's the apical surface right there. And so these upper cells are more cuboidal. They're certainly not flat like squamous, and they're certainly not tall like columnar. So even though they're not perfect little cubes, of the three choices, they're definitely cuboidal. And you can see deep to those cells with their big round nuclei, you have a more flat cell below it. So it doesn't matter the shape of the deeper layer. We're only naming it by the apical layer. So this is going to be a stratified cuboidal epithelium. Okay. And then here are two other examples. So I suggest you pause it and take a look at these and make sure you understand why both of these structures are considered stratified cuboidal epithelium. So all right, so coming back, we see this type of epithelium for secreting and absorbing, because oftentimes we'll have microvilli to go with that. We're going to have it in our sweat glands. I think that's what a picture is on the left, as well as in our ovary and in ducts in our testicles. And so actually the picture in the middle is an ovarian follicle, and the one on the right actually is a duct in the testes when it has a little sperm there in the lumen of it. So I took one example from each. Moving on out of cuboidal epithelium, what we have next is going to be columnar epithelium. So starting with a simple columnar epithelium, it's a tall cell with either a round or no oval nucleus in the bottom half of the cell for absorbing and secreting. So here are two examples. The upper example is showing that the nucleus is round, and the bottom example in pink is showing one with an oval nucleus. And as you can see, the round ones actually in this example are closer to the bottom, whereas the oval ones are closer to the middle. But you notice neither one of them has a nucleus in the upper half of those columnar cells. So these can have microvilli, and some of these can have cilia. I think you clearly can see the cilia in this upper photo micrograph that I showed. We're going to have these in our GI tract or respiratory tract, our reproductive tracts. We do have a stratified columnar epithelium. It can be two columnar cells on top of each other, but it's much more likely to be shorter cells with a columnar layer on the surface. Absorbing and secreting against. Some places are going to have cilia. If you look at the two photo micrographs, the one on the bottom in the purple, this one has cilia on the surface, whereas the one up above with the blue nuclei does not have cilia. Okay, it's not common, so I'm not giving you normal locations for this. There's only a couple of places that this happens, like the inside, the conjunctiva inside your eyelid, and then part of the male urinary tract. So it's a very rare epithelium. Now, don't forget, we had the great student fake out, the pseudostratified columnar epithelium, where the tallest of the cells are going to reach the apical surface, and it's always ciliated. So here are two photo micrographs of it. You can see the cilia on the surface of both of them. At first glance, you go, oh my God, there's nuclei all over the place, but compare those pictures to these pictures. Do you see how on this one, on the apical layer, the nuclei are all pretty much at the same level, and you can see that every cell has a nucleus on that. Whereas on this one, you cannot see that. The nuclei are all over the place. Okay. So, pseudostratified columnar epithelium almost always secretes mucus, and that is courtesy of those goblet cells. So the picture on the left where you have the blue arrow, you can see the cell here that's paler, and there's another one there, and it's another one there. So just think a little tall, skinny champagne goblet. That's how it got its name. Now there is one more type of epithelium I have not alluded to previously, and that is something called the transitional epithelium. And it's called transitional because it changes the way it looks, depending on how stretched the epithelium is. At first glance, when you look at it, whether you're looking at the one on the left, which is not stretched, versus the one on the right, which is stretched, you would say, maybe it's a stratified cuboidal, okay? And in fact, when you look at the cartoon drawing, you go, yeah, definitely a stratified cuboidal. But here's the difference. Look at those apical cells. Do you see how those apical cells are bridging over the tops of more than just one cell? Whereas in a true stratification, they're on top of each other. So those apical cells are definitely a lot wider than the thickness of the cells below them. And so we call these umbrella cells, okay? So I put some more drawings here to show you those umbrella cells, okay? And when they are stretched, what happens is everything gets to stretch out because this is lining the lumen, for instance, of your urinary bladder. And overnight when you're sleeping, your bladder is stretching, stretching, stretching. And so what happens is these cells, the umbrella cells, get to flatten out. And so instead of looking like it has maybe 6 to 10 layers, all of a sudden it looks like it only has maybe 3 to 5 different layers, because the transitional epithelium allows it to flatten without pulling the cells apart. And with that, we're done with epithelium, which means we're done with everything through tissue. So I want to thank you for watching these, taking notes, continuing with your studying, and continue to work hard. And I will see everybody in class. Have a great rest of your weekend.